Hello everyone. Today's video is all about cellular transport, basically how things move in and out of a cell. And our specific objectives are to discuss the structure and function of membranes in living organisms, because that is how things move in and out of your cell, and describe how materials are exchanged across those membranes. So to start off with, a few things that a cell needs. Why does cellular transport have to actually happen? Is because cells need to take in water and sometimes have water leave them. They need to maintain the correct balance of ions, which also includes water balance, nutrients to come in, sugars and amino acids and all those good things that you eat have to then get into your cells, and some hormones need to get in and out of your cell depending on what that cell needs and is making, and waste must exit the cell. So think of a whole human. What does a whole human need? And it's very similar to what a cell needs. So how things move in and out, as I said before, is through the cell membrane. So to explain the very basics of a cell membrane, the cell membrane is made up of phospholipids with proteins put throughout it and cholesterol, and is known through the term the fluid mosaic model. And that is because it's fluid, meaning it's moving, it's not static. It doesn't just stay, it's not like a wall that is a solid structure, it's constantly moving. And mosaic means that it's made up of more than one item. So it's not just one complete thing, it's made up of a whole bunch of different things. And as I said, those different things are your proteins and your phospholipids and your cholesterols. And the proteins can be two different types. The glycoproteins are the proteins that generally have carbohydrates attached to them, and those kind of signal what type of cell it is. So each person has an individual type of cell, and that's why organ transplants kind of get tricky. But that's what the glycoprotein is, to say, hey, I'm Johnny's cell, hey, I'm Jimmy's cell. And the transport proteins are proteins that are in there that help things move in and out of that cell membrane. And the cholesterol is actually important, kind of as a binding force, and to make sure that the phospholipids and proteins can constantly move around. But in general, the cell membrane's job is to regulate what goes in and out of your cell. And therefore, it has your hydrophilic side and your hydrophobic side. So a phospholipid, if you remember, has a hydrophilic end and a hydrophobic end. The hydrophilic end is on the outside, the hydrophobic end is on the inside. And so you'll look at the picture and see the little circles are the hydrophilic side of a phospholipid, and the fatty acid chains at the bottoms, those two lines coming off of it, are your hydrophobic ends. And that's going to make it not be completely permeable, it's semi-permeable. So all of these different things allow the cell membrane to actually determine what comes in and out. It's not just an open screen. Things don't just willy-nilly come in and out of it. So they move through depending on their size, whether they're hydrophobic, whether they're polar or nonpolar. And that's what makes it selectively permeable. And things that can go through are your hydrophobic nonpolar molecules, like your oxygen, carbon dioxide, and nitrogen, and some small uncharged polar molecules like your waters, glycerols, and ethanols. Although those do need a little help to go through, but they can. So think of those as like the things in the air that can go through the screens in your windows, whereas there are certain things, like bigger things generally, that cannot go through that screen. And those things get denied without extra help. And those are your larger molecules, specifically your uncharged polar molecules like your amino acids and glucose, and your ions. So if anything is charged, it cannot just go in and out. So this way, the cell membrane can actually determine what it can take in and what to keep out and not go through. And that is also determined by your transport proteins. So we'll get into that in a little bit. So because of those differences, you need a few different types of transport. And the two general types are passive and active. And there are different types of passive transport: diffusion, osmosis, and facilitated diffusion. Um, and two general types of active transport. They're known as bulk transport, endo and exocytosis, and active transport is also just kind of a general way that things move in and out using transport proteins. So we're going to go through each one of these and how they move things in and out and how they differ and why they are needed by the cell, all these different types. So the first type is diffusion. Diffusion is a very basic term that can be talked about without a cell membrane involved, but diffusion is things moving, molecules moving from a high concentration to a low concentration. So they move because of a concentration gradient. And this is necessary for a lot of cell processes we'll discuss in other videos, like cell respiration, photosynthesis. They're often used to actually then produce energy. And just in general, think of things like smells moving through a room. They are all concentrated in one area and then they spread out. But if you have a concentration gradient, things will move until they end up with equal concentrations. So you'll see in the picture, your cell membrane, the membrane, gray items, are blocking the molecules of dye, but because the molecules of dye are small enough to actually move, they will move until they reach equilibrium. Same thing if you have more than one type of solute. Now, once they reach equilibrium, it does not mean that things stop moving across. They just move in equal amounts. So if two molecules move from left to right, then two molecules will move from right to left. So it's just kind of an equal flow of things. It's not flowing in one direction versus the other. So think of diffusion as going with the flow of students in the hallway. Now, osmosis is simply diffusion with water. And this one gets a little confusing because it is high to low water concentration. And we often do not talk about solutions in terms of the amount of water. We usually talk about solutions in terms of the amount of solute. So that's where it gets a little tricky. But if you look at the picture, you'll see on the left-hand side, there's a lot more water molecules. And so the net movement of water will move from the more water molecules to the fewer water molecules. And this is generally when the solute cannot move across the membrane. And this is actually how things in your body can get hydrated versus dehydrated. So it becomes a very big part of your body. Whereas diffusion is how, say, oxygen gets into your bloodstream. It's just you have lots of oxygen in your lungs and less oxygen in your blood, so it diffuses from high concentration in your lungs to low concentration in your blood. Water in your kidneys, for instance, and just in cells in general, are going to move in and out of cells and hydrate versus dehydrate. So there are different types of solutions in terms of osmosis and water concentrations, and they're known as hyper, hypo, and isotonic. And the name actually tells you what they mean. Hyper means over, tonic is solute. So hypertonic is when the solute concentration is higher, meaning there's less water. So water always moves towards hypertonic solutions. So because it's has over solute in it, has too much solute. So cells in a hypertonic solution will shrink because the water will move out of them. And this doesn't necessarily have to relate to a cell, but think of it in terms of a cell. It could just be across the membrane, but notice in the picture you have more salt outside than inside, so that is known as a hypertonic solution, and the water will move from inside the cell to outside the cell. In a hypotonic solution, hypo means under, solute is tonic, like before. And so the solute concentration is lower, so there's more water. So water moves away from hypotonic solutions. So when a cell is in a hypotonic solution, the cell will swell because the water will move from the outside to the inside. So the way you can look at the picture is in two ways. You can say, okay, I see that this is a hypotonic solution because outside there's less solute than inside, so the water is going to move in. You can also change that 10% salt to 90% water and that 20% salt to 80% water and say, oh, the water is going to move from 90% to 80% because the water, remember, is still moving from high to low concentration. That's a big thing in passive transport. No energy is used. Water is going to move from high to low concentration of water. Now, if you look at this picture as well, you can say the solution is hypotonic, the cell is hypertonic, the inside of the cell. So you actually have two solutions in each situation. Inside the cell is hypertonic because it has more solute than the outside. The outside is hypo, the inside is hyper. So water is always going to move from hypo to hyper. So you can also think of it in those terms. So always from hypo, under to hyper, over. And then the last solution is isotonic, meaning you have equal concentrations and water will move equally inside and outside. So the size of the cell does not change. So in terms of the other two, hyper, the cell is going to shrink, and that's why in certain solutions, if you are trying to, say, give somebody a saline solution, you don't just give them an IV of 100% water, because if you give them 100% water, you're going to put their blood cells in a hypotonic solution, and the blood cells will swell and explode. But if you put them in a isotonic solution, then nothing will actually happen to them. So that's actually what happens to different cells. And if they are put in a solution, a hypotonic solution, they're going to swell and they're going to burst. And that's known as lysis if you're an animal cell. In a plant cell, because plant cells have cell walls, that's actually just known as turgid. There's turgor pressure. The cell membrane is putting pressure on that cell wall. And that actually is a normal plant cell, and that's what makes plant cells more rigid. So if you think of when you go into a grocery store and they're constantly spraying water onto the lettuce, it's so that it doesn't look wilted. It's so that the cells are turgid. Now, there's the other end where you put the cell in a hypertonic solution and it shrivels. And an animal cell, not that big a deal. You can rehydrate that cell. But in a plant cell, it's known as plasmolysis. It's plasmolyzed. And the cell membrane kind of shrinks. And that's what happens when your lettuce looks wilted. So think of if you've ever had a salad, you put salad dressing on it and you have it sit there for a while. Say the next day. Your lettuce looks like it's actually wet. It looks like it's taken in the salad dressing, but it's actually the opposite. The water has left the salad dressing because the salad dressing had more solute in it than the cell. So the water left the cell and the cell membrane shrunk and it kind of got a little like wilted, and your lettuce actually looks wilted. It's not actually wet. It's not, doesn't have extra water in it or extra salad dressing. And if you do look closely at the salad dressing, it is diluted. More water has left and come into it. So that's what could actually happen to your cells inside your body if you get injected with a solution that is not isotonic. So it is very, very important before doctors give you an IV of a solution, they have a general sense of what normal humans cells are made up of, but they have to double check and make sure that they are giving you an isotonic solution, or else you will overhydrate or dehydrate, which is another issue. If you actually drink too much water, then you could cause problems internally, because then you would have too much water going into your cells. So that could cause even bigger problems, and that's why having electrolyte drinks is important. So then the next type of transport is facilitated diffusion. This is diffusion that uses proteins but still doesn't use energy. These are kind of like doors, but doors that don't need energy to open. So say we just have a door in our classroom that certain people can fit in. So you have to be a certain height to fit through this door. If you're too tall or too short, you don't fit. And that's what these protein channels are for. And these are used for a lot of larger molecules and many ions. And ions are specifically, the proteins are known as ion channels. And so it's basically a specific shape that allows that specific molecule through. So you'd have a sodium channel, you'd have potassium channel, you could have a glucose channel, you could have kind of any molecule channel, and things will move through them. They don't move through as smoothly as they move through diffusion, because they only have specific places. It's like if you all got up and tried to go out the door, you're not as efficient if you could go through the wall and the door. If you could go through the wall and the door, everyone would get out in a lot quicker manner. So those are the three types of passive transport. And a few things that could go wrong with those is specifically an example known as cystic fibrosis. So cystic fibrosis is when mucus builds up in your respiratory tract, and you have a hard time breathing, and it gets into your lungs. And the reason for that is in cystic fibrosis, you have a gene mutation that messes with your chloride ion channels. So chloride ions can't get through. And when chloride ions can't get through, then water doesn't move. So if you look at the picture outside the cell, if you have chloride ions that are able to move through the outside of the cell, that changes the solute concentration, and therefore water will also follow the chloride ions. But if chloride ions can't get through, no water will move out, and this mucus will build up. And that makes it really hard to breathe. So that's a problem that you could have if you don't have chloride ion, and it affects facilitated diffusion and osmosis. So now the next general type is active transport, which is a type of transport that uses energy. Passive does not use any energy. And it uses energy because it's moving things from low concentration to high concentration. So if you think of it in terms of moving in the hallway, if you're trying to move backwards away from everyone else, you're trying to swim upstream, it's a lot harder. And one general type of bulk transport or active transport is called bulk transport, and the two types are endo and exocytosis. And these are for moving large molecules. And endocytosis is taking in those large particles, and exocytosis is removing them. And there are actually two different types of endocytosis, pinocytosis and phagocytosis. Pinocytosis is moving fluids, phagocytosis is moving foods. And I always think of phago, even though it does not begin with F, it sounds like it does. So phagos is foods, so solid things. So if you're trying to take in a group of water, then it's pinocytosis. But if you're trying to take in a glucose molecule, that would be phagocytosis. So this picture shows the process. Essentially for endocytosis, the cell membrane creates this little pocket for molecules to come in, and then it creates that vesicle inside. And for exocytosis, you have that vesicle, and it merges with the cell membrane and releases them. So that's often how hormones will get in and out of your cells, and just your larger molecules. Or if you just need to get rid of a huge amount at once, like your teardrop is actually through exocytosis. So you have a vesicle of fluid, and it moves out through exocytosis and produces that teardrop. And that applies to the cell in many different ways, but a major example is your white blood cells are there to kill bacteria. So the way they do it is actually to um, engulf that bacteria through endocytosis. And then what happens to that little vesicle is it merges with lysosomes that, or a lysosome that has enzymes, digestive enzymes, breaks it down, kills the microbacteria, and then it actually gets expelled through exocytosis. So that's just one example. Another is your ribosomes make a protein that then in your Golgi apparatus gets packaged, etc., creates this little vesicle, and that protein gets sent out through exocytosis to go and do its job elsewhere if it's not necessary in the cell. And then active transport that is not bulk transport is done through proteins, and it's, as I said, using energy low to high. And the reason this one is actually done, because you're like, well, if it's using energy, why are we doing it? Is sometimes you actually need to build up a concentration gradient. And this is huge in the neurons. Your nervous system uses electricity to move things through your body. So you need to build up a concentration gradient, kind of like a battery, like you're charging your battery. So that's how active transport is used, is to move things from low to high concentration to make that concentration gradient. And the example, as I was saying, in the nerve cell is using a sodium-potassium pump that pumps, if you look at this picture, it pumps your 3 sodium ions out to 2 potassiums in. It just it's literally like a pump. And it uses this energy because what it's doing is going to build up your sodium ions on the outside and potassium ions on the inside. So your nerve cell, as I said, gets charged up like a battery so that when it needs to send information, it's connected like a wire and can move that electricity, and then has to remake that gradient. And it's an electric gradient because the ions are charged. If the ions weren't charged, it would just be simply a concentration gradient. So to summarize everything, the two general types of transport are passive and active. Passive goes from high to low, uses no energy, and can move either through the cell membrane or proteins in the cell membrane. So the diffusion osmosis use the phospholipids, and the facilitated diffusion uses the proteins. And then for active, energy is used, and it's generally moved from low to high concentration. And the type that uses the cell membrane fully is bulk transport, the endo and exocytosis. And the proteins, those proteins are known as pumps, whereas the proteins for facilitated diffusion are generally transport channels. Kind of like you have doors that anybody can come through, and then pumps that you have to be pushed in and out of for those. So that summarizes all the different types of transport that your cell uses. And as you see, your cell does need all those different types in order to be able to do all of its jobs. So I hope that was useful, and you can now start looking at how that actually applies to specific molecules within your cell and where they would be useful depending on the type of cell, if you're a muscle cell or heart cell or stomach cell or all the different types of cells.